Hello everyone, today we will be discussing the fate of amino group in the metabolism of amino acids. So we know that all amino acids have the amino group. So this amino group is transferred to another keto acid and that step is transamination. So transamination is the transfer of amino group from one amino acid to a keto acid and it forms another amino acid and a keto acid. Okay. So if you take an example of a simple alanine, so this amino group is transferred to a keto acid. For example, alpha ketoglutarate. So this is transferred to alpha ketoglutarate to form another amino acid. So this amino group is transferred to this place. So we will get another amino acid. Which is the glutamic acid plus another keto group. That is, the keto group will be transferred to the first amino acid. So what we will get? So we will get pyruvate. So that is why only there is a transfer takes place. This amino group is transferred to the keto group and another amino acid and another keto group is formed. So you will get glutamic acid and pyruvate. Okay. So what we require, the coenzyme is a pyridoxal phosphate, PLP. Pyridoxal phosphate is a coenzyme involved in the transamination reaction. So all the transamination reactions we require pyridoxal phosphate. So now we will see the common enzyme is here. So since alanine is transferred, it is called as alanine transaminase. ALT is the enzyme involved in this transamination reaction. So in alanine transaminase, the amino group of alanine is transferred to the alpha ketoglutarate to form glutamic acid and pyruvate. That is, amino group from one amino acid is transferred to a keto acid to form another amino acid and a keto acid. So this is simple terms of transamination. Now we will see the mechanism. So we know that pyridoxal phosphate is involved. So what happens is, so from one amino acid, the amino group, it is transferred to a pyridoxal form, pyridoxal, and it forms pyridoxamine. That is, amino group is transferred to the pyridoxamine. So that this amino acid will become the keto acid. Okay, and the reverse reaction, that is, in the first step, the amino group of the amino acid is transferred to the pyridoxal, the aldehyde form, to form the pyridoxamine. Then it is transferred to, then this amino group is again transferred to the keto acid to form the second amino acid. Okay, and then pyridoxal phosphate is regenerated. Okay, group to the alpha ketoglutarate. So mostly the amino group is transferred to alpha ketoglutarate itself, and the enzyme is AST, aspartate transaminase, to form the oxaloacetate and glutamic acid. Oxaloacetate and glutamic acid. So that is the aspartate transaminase reaction. So with the help of this transamination, now what happens is that the amino group is transferred and what we get is glutamic acid. Now this glutamic acid has to undergo deamination to release the amino group as ammonia. So we usually detoxify amino group as ammonia, and this ammonia enters the urea cycle, and it is excreted as urine. So next step is deamination. So deamination is of two types, that is oxidative and non-oxidative. Oxidative as well as non-oxidative. So oxidative deamination, one example is glutamic acid. So glutamic acid, it undergoes oxidative deamination with the help of glutamate dehydrogenase enzyme. So this enzyme can utilize both NAD and NADH, NADP as coenzyme. Okay, so it can, so only one of the rarest enzyme which can utilize both NAD and NADP as its coenzyme. So glutamate, it undergoes dehydrogenation, and what is the compound that is formed? We will get ammonia. Ammonia is liberated plus alpha ketoglutarate is formed. Okay, so that is a deamination process, the glutamate dehydrogenase. This is also a reversible reaction. So this ammonia is liberated, and this ammonia enters the urea cycle. So deamination, one example of oxidative deamination is this glutamate dehydrogenase. Apart from that, we have the deamination by the L-amino acid oxidases, then D-amino acid oxidases. So L-amino acid oxidases, so L-amino acid oxidase, so that utilizes FMN as a coenzyme. So any L-amino acid oxidase, L-amino acid is first converted to an amino acid, L-amino acid, and then it forms the keto acid. And then it utilizes FMN. So FMN is converted to FMNH2. And thus it converts, it generates hydrogen peroxide, which is in the peroxisomes is acted upon by catalase, and it releases nascent oxygen. Okay, H2O2 generated is catalyzed by the catalase enzyme, acted upon by catalase enzyme and releases nascent oxygen. And similarly, there is D-amino acid oxidase. So D-amino acid oxidase can oxidize glycine, and also any unnatural D-amino acid is converted to the L-amino acid. So D-amino acid oxidases utilizes FAD as coenzyme. So that is a one peculiar feature of D-amino acid oxidase. So these are the other types of oxidative deamination. Also monoamine oxidase, a small amount of monoamine oxidase that also generates ammonia, okay, in the intestinal tract. So these are some examples for oxidative deamination. So oxidative deamination, glutamate dehydrogenases, it utilizes glutamate NAD or NADP. It can utilize NAD or NADP as its coenzyme. Then L-amino acid, it utilizes FMN as its coenzyme. D-amino acid oxidase utilizes FAD as coenzyme. So these are the types of oxidative deamination. Then second is a non-oxidative deamination. So non-oxidative deamination are mostly the self-hydrases or the dehydrases. So non-oxidative deamination, that is a dehydrases. Dehydrases acting on serine and threonine, and they generate ammonia. Both of them generate ammonia. So serine generates ammonia and pyruvate, and threonine generates ammonia plus alpha ketoglutarate. And also in the case of sulfur-containing amino acid, cysteine, homocysteine, they are acted upon by desulfhydrases. And then they release ammonia. So cysteine generates ammonia plus pyruvate. Similarly, histidine, it is acted upon by histidase enzyme, and it generates urocanic acid and ammonia. So these are some minor pathways of deamination. Okay, so non-oxidative deamination, they do not undergo oxidation, they directly undergo action by the dehydrase enzyme and release ammonia. Okay, all these release ammonia. These are only minor pathways of deamination. Also in the intestinal gut, by the action of bacterial putrefaction, ammonia is liberated. So all these generates ammonia. So this we have to dispose ammonia because ammonia is highly toxic to the brain. So disposal of ammonia. Now we will see how this ammonia is generated, now how it is disposed. So there is we will discuss the disposal of ammonia. So disposal of ammonia, the first step is the first line of defense is the trapping of ammonia. This ammonia is trapped as glutamine, especially in the brain, intestine and most tissues. Then it is transported. So transported from the brain and most other tissues is transported in the form of glutamine. From the skeletal muscles it is transported in the form of alanine. And then the final disposal. So disposal, the trapping of ammonia. So we know that all these amino acids generate glutamic acid. So glutamic acid, what does it do? It traps the ammonia as glutamine. So glutamic acid traps the ammonia. Ammonia is trapped as glutamine. And the enzyme involved is glutamine synthetase, which involves ATP. ATP is utilized. Okay. So ammonia amino group is added to the ammonia is added to glutamic acid and forms glutamine. Then with the help of glutaminase. So in the liver, so glutamine is a transport form and it reaches the liver. So in the liver, by the action of glutaminase, glutamic acid is regenerated and ammonia is liberated. Ammonia is liberated in the liver. So detoxification of ammonia occurs in the liver as a urea cycle. So this ammonia enters the urea cycle and the waste product is urea. Okay. So the transport form, it is in the form of, from most tissues it is glutamine and from the skeletal muscles alanine. Okay. This alanine will undergo transamination. Transamination forms glutamic acid. Then the glutamic acid will trap the ammonia as glutamine. Then in the liver, it undergoes action by the glutaminase enzyme and glutamic acid is regenerated. Then the final disposal. So final disposal in human beings, it is the ammonia is disposed as urea. So we are ureatelic. In the case of fishes, the ammonia is liberated. Ammonia is released as ammonotelic. Most of them are ammonotelic, and the birds, they excrete ammonia as uric acid. So they are uricotelic. So we humans are ureotelic, then fishes and they are ammonotelic, and birds and reptiles are uricotelic. Okay, so that is a final disposal of ammonia, and then the ammonia enters the urea cycle and finally forms urea, which is excreted. So the normal excretion is around 50 to 30 grams per day, and this excretion depends upon our diet and the rate of protein turnover. Okay, so that is it. So in summary, the transamination is transfer of amino group from one amino acid to another keto acid to from another amino acid and the keto acid. So this transamination occurs in almost all the tissues. But then after transamination, it has to undergo oxidative deamination, which occurs in the liver. So this transamination plus deamination, that is called as transdeamination. So transdeamination, so this is a coupled reaction. Transamination occurs in all the tissues of the body. The oxidative deamination, especially by the action of glutamate dehydrogenase, occurs mainly in the mitochondria of liver. The ammonia is liberated. Then the trapping of ammonia disposal, the ammonia is trapped as glutamine, and with the help of glutaminase enzyme, glutamic acid and ammonia is liberated in the liver, and this ammonia enters the urea cycle. So that is how ammonia is excreted from our body. Okay, because ammonia is highly toxic. But in the kidney cells, though ammonia is toxic, we use it for the buffering action. Okay, by the action of glutaminase, ammonia is released from the glutamine, and we use for the buffering, for the acid-base balance. Okay, so that's about the transamination. In the next video, we will see the urea cycle and disorder.